And this meeting is being recorded. So welcome everyone to this session on pioneering in cancer immunotherapy here at Dartmouth. My name is Jake Rader. I'm going to be your moderator for the session. I'm going to start with a 1-minute introduction to immunotherapy itself. So we have a bit of a foundation. We'll do a very brief housekeeping, and then I'll get you to the meat of this, which is really a discussion with our three panelists, Professors Paul Guyre, Randy Noelle, and Margie Ackerman. And so without further ado, how does immunotherapy fit in with other therapeutic modalities? People have been treating cancer by surgery, by essentially cutting it out, since about 400 BC. And even today, surgery is a remarkably effective form when you can reach the tumor. Radiotherapy is a little over 100 years old, and again has a long established foundational presence in cancer therapy. Chemotherapy came along a little bit later, and again has been, at least what I grew up hearing about. Immunotherapy is much newer, but has quickly established itself as one of the pillars of cancer treatment, and indeed treatment of many other diseases as well. The main difference here is that the exquisite specificity of our own immune systems can be brought to bear against tumors for the benefit of the patient. And this can be done in combination with some of the older, more traditional modalities, or can be a standalone as well. And so immunotherapy has been part of Dartmouth's research and translational activities for a very long time, since before it was called immunotherapy. And so you'll hear a little bit about that today from our panelists. So before we jump into that, let me just get to a few housekeeping items here. I'm going to stop my share. And the first thing is that, as you've heard, we are recording this session. If you have any technical needs, please post them in the chat box. If you have any questions for us, there's something called Q&A, the question and answer box. Please use that. And if you see a question from someone else that you like or you'd like the answer to, you can upvote it by clicking the thumbs up icon next to it. And so very briefly, Paul and Randy and Margie are here today from the medical school and from the Thayer School of Engineering. I'll provide a brief introduction to each with a kickoff question, and then we will look forward to fielding your questions thereafter. And so let me start with Paul. Paul has been a professor of physiology and microbiology and immunology at Dartmouth Medical School since 1981. He's also a co-founder of Medarex, which is the most important immunotherapy company, bar none. He is also vice president of R&D at Celdara Medical, which is another Dartmouth spinout with a record of bringing innovation to the clinic. And so, Paul, I'll begin with you. As far too few people know, you spun Medarex out of Dartmouth long before immunotherapy was a thing. The impact of Medarex on medicine has been truly unique. Almost half of all approved human monoclonal antibodies came from Medarex's platform. The first approved checkpoint inhibitor, now called Yervoy, as well as two more recently approved drugs, Opdivo and Zimplava, also came out of Medarex. And the result of that has been thousands of lives saved. The 2018 Nobel Prize would not have been awarded the way it was without Medarex. And immunotherapy itself, the reason we're all here today, which is probably the most promising new field in medicine. And so there's few stories like this, and it all started here at Dartmouth. Can you give us this sort of 30-year epic in just a few minutes?
Well, thanks, Jake. I'd like to first give a shout out to my mentor and Medarex co-founder, Mike Fanger, and his wife, Sharon. We started Medarex when I was a young assistant professor, and Mike took care of all the business-related stuff. I have Sharon to thank for my early success in getting NIH grants. As Steve Leach has noted, the Norris Cotton Cancer Center is the only comprehensive cancer center not in a major metropolitan area. I didn't see my MD colleagues at work only. We had kids in the same class and on the same ski team. Thanks to cancer center clinicians, Medarex had the first ever bispecific antibodies in clinical trials right here at Dartmouth Hitchcock. So a big thanks to Lionel Lewis, Letha Mills, Pamela Ely, Frank Vallone, Peter Kaufman, and other clinicians who enabled our bispecifics to get into the clinic. Back then, bispecifics were essentially glued together and took a year to make, impossible to do in large batches under GMP conditions. Now a Medarex spin-out, Genmab, can make clinical grade bispecifics as well as 6-sided antibodies called hexabodies in under a month. Advancing immunotherapy further will take teamwork, and I don't think any place is better equipped for that than the Norris Cotton Cancer Center. Thank you.
Thanks, Paul. Yeah, it really is all about the people, and you've been one of them for a long time. Another one is Randy Noelle, who's here. Randy is a professor of microbiology and immunology and the inaugural holder of the Thomas S. Kosasa MD professorship at the Geisel School of Medicine. A cellular and molecular immunologist, Randy has made numerous discoveries that are being advanced for the treatment of cancer and autoimmune disease. He's also a co-founder of Immunext, another Dartmouth spinout that develops immunoregulatory drugs. So thanks for being here as well, Randy. And you're one of the reasons that Dartmouth continues to punch so far above its weight. Can you give us an overview of the most important discoveries you've made as a professor and your experience translating those innovations as an entrepreneur?
Thanks. Thanks, Jake. But let me preface the answer the following way. We all know our immune system is our guardian against diseases caused by any pathogen, including a whole host of viruses and bacteria. We know that all too well this year with the COVID plague that we deal with. However, particularly as we age, the immune system begins to falter and loses its ability to decide what's a threat and what's not a threat. Failure to recognize these threats culminate in the insidious development of cancer as we age. Cancers can grow unrecognized by the immune system with no attack or killing it, simply because it doesn't recognize it. And one class of drugs that we're talking about today are new immunotherapeutic drugs that can make your immune system recognize this cancer. In contrast, sometimes the immune system loses its self-control and lashes out against the host. The immune system unleashed, as it were. This results in the development of rampant inflammation and the development of autoimmune diseases that we all know all too well: rheumatoid arthritis, lupus, multiple sclerosis, and virtually all the inflammatory diseases that we suffer again as we age. Molecular immunologists like myself, like Paul, like Jake, like Margie, we look for triggers, molecular triggers within the immune system that we can target to restore the ability of the immune system to respond to cancer, or a molecular reset button to fix the dysregulation that an aging immune system has developed and has gone awry. So with respect to your question, at Dartmouth, we discovered a big red reset button within the immune system. This reset switch for the immune system, we targeted with a drug that we believed that this drug or this antibody could reset the immune system in individuals with uncontrolled autoimmune disease, MS, RA, etc. The drug is an antibody. It's to a molecule called CD154, and it entered the clinic actually quite a while ago, but posed safety concerns. And these are the trials and tribulations with developing drugs. Entered the trial, we were all very excited about it, but it posed very serious safety concerns. And in fact, the efforts were disbanded. Convinced that this was a one-of-the-kind opportunity and a one-of-a-kind drug, we started the company you referred to as Immunext, a Dartmouth-founded company. And over the next 10 years, we retooled this drug to make it safe and effective. And actually last year, this drug entered phase 1 trials, proved it was safe, and is now presently in phase 2 trials in humans to test if it can treat autoimmune diseases like multiple sclerosis, lupus, and rheumatoid arthritis. And we're really very excited about it. The number two molecule that we were been involved with about 10 years ago, smart people in my lab, smart scientists in my lab at Dartmouth discovered an immune molecule that we called VISTA. We learned that VISTA was a molecule within the immune system that prevented your immune system from mounting an attack against tumors. Immunext produced a clinical grade anti-VISTA antibody that blocked VISTA function, and as a result, enhanced the immune response to cancers and increased host survival in preclinical models in mice that we moved forward on. The clinical grade anti-human VISTA antibody developed by Immunext is actually now in phase 1 trials at the Norris Cotton Cancer Center and other cancer centers around the US to ensure that it's safe, and hopefully we'll see some evidence of its therapeutic efficacy. If I could just close with saying that the successes that you've heard about and you will hear about are the result of a remarkable scientific and entrepreneurial environment. That which you spoke about, those successes here are the result of a faculty of scientists that create an absolutely superlative intellectual environment where interaction and knowledge are freely shared. World-class graduate students, world-class postdoctoral fellows are really the engines that drive our successes. Translation of these discoveries from the academic sector to the commercial and clinical arenas is always a challenge and has been. However, Dartmouth, through organizations like the Dartmouth Entrepreneurial Network and local entrepreneurs interested in drug development, like yourself, Jake, provide a unique environment so inexperienced academicians like me can begin the long journey of bringing their discoveries to cure human disease. All of our successes are the result of this robust community in the Upper Valley that's been built over the last 2 decades. And this community is going to serve us all very well for future Dartmouth scientists and entrepreneurs to really move Dartmouth-based discoveries into the clinic. So that's the story.
Well, thank you, Randy. And, I just want to highlight a couple comments that you made. We work with universities all around the world, and what's going on right now at Dartmouth is not normal, right? There is that, as you say, of high quality brains, high quality humans. And now there is something that wasn't there when Medarex was started, which is this ecosystem of translational capability as well. And so it's a very exciting time to be here. Thank you for those comments. Margie, I want to get you into this conversation. Margie Ackerman is a professor of engineering at Dartmouth's Geisel School of Engineering, where she leads interdisciplinary research at the interface of biomedical and engineering sciences, developing high throughput tools to evaluate antibody response in disease states ranging from infection to cancer in order to aid the therapeutic antibody and vaccine design and development. And so, Margie, some think of immunotherapy as the exclusive domain of medical schools, but you're an engineer. Can you describe what engineering brings to medicine, how that's done at Dartmouth, and some of your specific areas of interest?
Yeah, so I think we bring some really sharp pencils, like Randy alluded to already. The intersection between engineering and immunology is such an exciting place to be at Dartmouth. I think, Thayer has plans to double, and along the way, we have been investing heavily in this space. I think we have some key strengths in molecular optimization and the ability to turn insights into novel interventions. We've been motivated by the success of faculty companies such as Adimab, which is a premier antibody discovery technology company started by engineering faculty member Tillman Gerngross and still located in the Upper Valley, right across from the hospital, which has put hundreds, if not thousands, of antibodies into the clinic or into preclinical models through corporate partners. as well as colleagues in the Centers for Imaging Medicine and Surgical Innovation who are developing new instruments, new molecules, and new analysis capabilities to define things like tumor metabolism and the effects of radiation and immunotherapies in vivo on patients' tumors. From my personal perspective, the impact of engineering on immunotherapy really runs from soup to nuts, from identifying and optimizing targets and targeting agents to collating clinical data and learning from it to define new avenues for personalized medicine. So my lab works mostly at the beginning end of that continuum, using directed evolution and protein engineering methods to screen billions of different protein variants, billions of different anti-VISTA antibodies potentially, for rare versions with enhanced tumor recognition or enhanced activities in vivo or in vitro. And one thing that's been particularly exciting is to see that a number of students from my research group participate in Dartmouth's PhD in Innovation program, where they've been trained in entrepreneurship and innovation and are applying that skill set to address problems in cancer therapy.
Fantastic. Thank you, Margie. And just the sheer productivity of the Thayer School in this field has been astounding. And so thank you for that and your colleagues as well. And so now I'd like to open it up to the audience. We've got 250 people here today, and we're already, of course, starting to get some questions. I'll put the first one out maybe to Randy, and it's a question here about stage and immunotherapy. And so are immunotherapies more or less effective at different traditional cancer stagings?
Actually, yes. The drugs that are out there, the two main drugs that we're referring to that really were the basis for the Nobel Prize and the real transition in immunotherapy actually work when your immune system is at a different stage in attacking the tumor. One of the drugs, ipilimumab, makes more of your cells activated to see the tumor. And the second drug, nivolumab, makes those activated cells more efficient in killing the tumor. And so, yes, I mean, we are developing, the drugs that have developed work at particular stages of improving the immune response to the tumor.
Great, thank you. Thank you. I'm sorting through. So there's a question at the top of the list here now. What is Dartmouth's unique value add to this field? What's happening at Dartmouth that is not happening in other places? This is like a planted question. I love this one because there is so much and so much of it is going on in your labs. But let me guide that first to you, Margie. Do you want to speak about what's unique about Dartmouth vis-a-vis the rest of the world?
Yeah, I mean, I think a major part of it is growth, right? So we both have some runway and history that you've heard Paul and Randy mention, as well as being an institution that's expanding and growing. And that's an exciting place to be. That's something that not every institution can claim. I think another way Dartmouth has sought to differentiate itself is really lowering the barriers to faculty and student ability to practice their inventions and to translate them. So we give a lot of freedom to the bright minds, the sharp pencils that work here in order to both pursue their academic undertakings as students or faculty, as well as to push innovations into the marketplace.
Fantastic. And, you know, I'll add to that. Everything you're hearing about here is almost by definition unique to Dartmouth, because somebody like Paul or Randy or Margie, they invent something, they file a patent on it, and therefore that is something that they or their licensees are uniquely able to turn into a product. And so, you know, bispecific antibodies back in the 90s coming out of Medarex, that was first done, as Paul said, at Medarex. Vista, which is now one of a very small handful of checkpoint targets, that came out of Randy's lab, right? And these optimized structures that Margie's working on, these are unique, not just in academia, but in the universe, right? Nobody has ever seen the one out of the billion that she's able to pluck out of the soup that happens to work so much better than the rest of them. And so each one of these things is almost by definition unique. And what I'm most excited about is the unique ecosystem and the culture here, which I think is markedly different than what I see at other universities, which is that there's true collegiality here still. Yeah, everybody's competitive. Everybody wants to do well. They want to get their grants and get their papers published, but people help each other. And that's just part of the cloth here, which again, you don't see everywhere, and you don't see at many of the leading places as well. And that's a huge competitive advantage here, where everybody's throwing in to help each other on the best ideas that have the best chance to help patients. So lots of questions pouring in here. So let me get back to it. Can any of the three panelists speak to immunotherapy in breast cancer and the status of the treatment of breast cancer with immunotherapy? The assertion is they don't think it's quite as far along in breast cancer than other types. Any thoughts on that?
I think that in part reflects some difficulties in targeting solid tumors, which are drug delivery challenges. So if you have an amazingly effective molecule, you still have to get it to its site of action. And it's been more difficult to get antibodies or cellular therapies to certain tissue sites. And so I think there's a lot of promise in combining modalities, in addressing and taking novel strategies to localize those interventions to the sites where they need to act.
Yeah, thanks, Margie. I'm thinking specifically of Steve Feiring's lab at the Norris Cotton Cancer Center, which has done some really great preclinical work in collaboration with other academicians, specifically on breast cancer. But this is preclinical, it's early stage, but it recently received a lot of press. Randy, you've got...
Yeah, so I was just going to say, just for perspective, what we've learned over the last 6 or 7 years is that we have now a platform of immunotherapy based on 2 drugs. And depending upon which tumor type you look at, it's a phenomenal success. I mean, response rates 30, 40%, which is astronomical for new drugs in cancer. But now the challenge is to move from 30 to 40% to 90%. And that's the challenge now. What additional targets are we going to add to that platform to improve efficacy in a wide range of solid cancers? And breast is one of those where there's not as much success with current immunotherapeutic approaches. But certainly the wisdom is that when we add on additional antibodies or other therapeutics onto that platform, we'll see much improved success across all human tumors.
Yeah,
there's a lot of room for using, machine learning approaches to identify
Yeah.
the patients who are most likely to benefit from a given intervention. So all of the trials that don't work as well as hoped or work for a subset of people, there's a big effort that's aimed at understanding what's different about one person's tumor or one individual from the other that might better inform future treatment options.
Yeah, and again, to emphasize what Margie just said, this is all going, or it is all becoming personalized. You will go in and you will be screened and assessed, and your treatment may be very different from a person with exactly the same tumor, but looks molecularly different than you are. And so that's the future of that we will be living with.
Yeah, well, you can tell it's a good question when the panelists keep going back and forth without any prompting from me. So fantastic question. And precision oncology absolutely fits into this. The first slide was adapted from another local oncology company, Javelin Oncology, that's doing exactly that, matching patients to therapies. There's one more question I want to get to. I know we're getting to the end of our time here. Paul, do you want to speak to the ability of cancers to develop resistance to immunotherapies?
Yeah, well, we're learning a lot again because of molecular techniques. What leads to resistance? A big part of the resistance is having molecules either on the tumor cell or on adjacent cells that block the checkpoint antibodies or block the immunotherapeutic mechanism. There's particular types of macrophages, the big eater cells of our body that protect us against bacteria, particular kind that differentiate into macrophages that will suppress the T cells locally. And so, again, these are things that we've only learned in the last few years, but we're now seeing, okay, these called M2 macrophages, we just have to regulate them, make them go back into M1 macrophages, the kind that kill bacteria and also support T cells to kill the tumor cells.
You can kind of flip that question on its head too, right? Because one of the really special things about immunotherapy is it can have a vaccine-like effect, and that patients can be immunized against their tumor, and that response becomes independent of the drug and an inherent new capacity that their immune system has. So both outcomes can happen. Tumors can become resistant, or the immune system can really get trained to clear tumor in a way that's unique and goes beyond the drug's capabilities.
Fantastic. Thank you, panelists. We're coming near the end of the time here, and we've got so many really well-informed questions here. I'm going to ask the organizers if maybe we could capture these questions and send written responses to them after the Zoom webinar. Hopefully that'll be technically possible, and we'll get you some responses. But the very last, the top of the question list here that's been upvoted and upvoted, it brings us back in such a nice full circle. Randy, I'm going to send this one to you. How does the VISTA antibody differ in function from the ipilimumab antibody that was, it says developed by Jim Allison, but in reality it was developed by that Dartmouth spin-out that Paul co-founded. Jim, of course, identified it originally, but then that went through translation through Medarex. And so, Randy, why don't you tell us some of the similarities and differences between ipilimumab and VISTA?
So, the current immunotherapies that we have today, ipilimumab and nivolumab, and we just briefly talked about it, they improve the activity of activated T cells. 95% of our immune system is sitting around waiting to do something, doing absolutely nothing until somebody attacks you. They're just sitting there waiting. Ipilimumab and nivolumab adjust once the immune system is activated and triggered to mount a response against a tumor. VISTA, in fact, targets all of those resting guys to educate them that there is a tumor there, get off your butt and start moving. And so it really improves the response to tumors from this massive resting population of cells to move them into the activated population to enhance tumor surveillance.
Fantastic. And we are fresh out of time here. I'm going to put up one more slide so that people can reach out, and they have the appropriate contact information as soon as I can find it. There we go. And this is a thank you to everybody. Fantastic engagement, fantastic questions. I think next time we know we'll schedule this for 6 or 8 hours on a Zoom meeting. But we will try to capture all of these questions and get you some answers. Thank you very much for joining us. And I look forward to the next one.
Thank you all.
Thanks, everyone.